Thank you for joining us in our presentation on bringing innovation to teacher education and surveillance studies to Eastern Tennessee. My name is Becky Trout-Rexell, and I'm an associate professor in the Department of Entomology and Plant Pathology. And my co-authors and co-speakers today are Michael Campanovo and Corey Day, who will be coming in the roughly 5 and 10 minutes to talk to you about their roles with this project. This project is really focused about understanding lacrosse encephalitis, which is a mosquito-borne disease. The virus typically infects children under the age of 15 years old, and it presents within 5 to 15 days as a summertime illness. Children are typically finding themselves with fever, headache, nausea, and or vomiting. And in certain situations, the virus can actually become neuroinvasive. And in this case, children present to the hospital with seizures, coma, encephalitis, and or cognitive disorders. As usual, there is a cost associated with infection of this virus, and it can range anywhere from $50,000 to over $3 million per case. One of the things I'd like to note as a part of this GIS symposium is that we do see specific clusters of where this lacrosse virus neuroinvasive disease occurs. We see many cases in Eastern Tennessee, Western North Carolina, and also in the Northern Appalachia region, although this virus was first identified in La Crosse, Wisconsin, up here. This virus is transmitted by a mosquito, the eastern treehole mosquito, which prefers to feed on these scurrid reservoirs and lay their eggs in these container habitats, which do include artificial containers and natural containers. We have noticed, though, a change in the epidemiology of lacrosse encephalitis, and this typically occurs after the introduction of the Asian tiger mosquito or the Asian bush mosquito into an area. And we hypothesize that it's very likely the presence of the Asian tiger mosquito that is increasing or enhancing lacrosse encephalitis in our area in Eastern Tennessee. I first started working on this virus and this disease ecology scenario when I received a phone call from the CDC indicating that a child from Maynardville, Tennessee, passed away from lacrosse virus. There was an autopsy that was done and that confirmed that the virus had indeed infected this child. On August 22nd, I was notified about this, and so I was asked to set traps around the area and identify the virus within the mosquito vectors. And we actually were able to do this. We were able to find the virus within eastern treehole mosquitoes that were actually very similar to the virus amplified from the child. And so we had this genetic match of this one lineage that these mosquitoes were indeed positive with lacrosse virus. One of the other things I noticed while we were working on this study was that the mosquito populations were not the same across space or across time. And so what I asked a spatial epidemiologist to work with me and try to identify if there were any clusters of these different mosquito vectors where there would be more in one area and less in another area. And we found these clusters of the Asian tiger mosquito and the eastern treehole mosquito. But what was really interesting was that when we overlapped or overlaid these two clusters, this is where we actually found lacrosse virus within the mosquito population. And there was actually another case of lacrosse encephalitis that occurred within this area. So there was something happening here with this, where these two mosquitoes were co-occurring. So I wanted to look closer at the fact that these mosquito vectors are ovipositing or laying their eggs together and what's happening with this larval competition. So I had an undergraduate, Drew here, who looked at this in a bit more detail with the help of the Knox County Health Department. We set out a number of these oviposition cups. They're really just black and white cups that we nail to trees or stake to the ground. And we found that there were a lot of the cups, actually a majority of them, had both the Asian tiger mosquito eggs and other eggs that were present within them, and that this association was significant. It wasn't a random chance that this occurred. We looked at this across space, and we found that, in some weeks we would have a site that had both mosquitoes co-occurring together, and in other sites we would find that there were no mosquitoes present within these cups. This was really interesting too, because it changed across not just across space, but also across time. And when we looked at this co-occurrence together across time, we noted that that's actually when we were seeing lacrosse encephalitis cases occurring within children as well. So then my next student, Devin Rowe, came in and worked on a master's project with me, trying to look at this idea of this connecting the mosquito populations to cases of lacrosse encephalitis. And so he did 20 weeks of mosquito surveillance across the county at a number of different sites. And he found, again, these population clusters where there's more of these mosquitoes present than likely to be there if it was done by random chance. And what was even more interesting is that where these mosquito populations co-occurred, in other words, where we had the Triceriatus or the eastern treehole mosquito populations overlapping with these Asian tiger mosquito populations, we actually had lacrosse encephalitis case occurring at these overlapping windows in time. And this actually was suggested in the literature back in 2008 when Dr. Bevins did larval competition studies where she had a bunch of cups and put known numbers of mosquitoes in each of the different cups and had these mosquitoes compete to become adults. And what she found was that when this competition occurred, the eastern treehole mosquitoes were likely, that this competition enhanced lacrosse virus transmission. So now that we have this idea where these two mosquito populations are co-occurring, that there's some kind of something is happening here that is leading to lacrosse encephalitis occurrence, to a lacrosse virus transmission to occur. And so what I needed to do was get the community involved and try something different, specifically because I can't be everywhere, my program can't be anywhere. And if we can get the community just aware of lacrosse encephalitis, I think that could actually help prevent transmission too. And so that's where our community engagement and workforce development project, which Michael Campanova will talk about here in a moment. Thank you.
Thanks, Becky. The goal of Megabites is to encourage students to learn more about and grow their interest in medical entomology and geospatial technology by engaging them in a citizen science project. Let me provide you with a brief program overview. We work directly with middle and high school science educators from East Tennessee over the course of about 9 months. In the summer, educators spend a week with us for an in-depth academy on mosquito life cycles and habitats, mosquito-borne diseases, GIS, and community engagement. Then we transition into the student-centered component of the project. At the start of the fall semester, educators and their students develop hypotheses they want to test. They set out mosquito egg traps and record data about the location and landscape characteristics around the traps using Survey 123. Mosquito eggs are collected once a week, every week, for 6 weeks. Eggs are then sent to the UTK Medical Entomology Lab at the end of the fall. Corey is going to give you the details in a few minutes, but at a high level, he and his team will raise the eggs to adulthood and identify them by species and gender, and then they are batch tested for lacrosse virus. This all happens over the winter. In February, we reconvene for one Saturday to review all the data with the educators. We practice building maps and apps with the data in ArcGIS Online. Inspired by Sean Anderson's webinar earlier this year, we even built dashboards and a hub to quickly and easily present the data and make it more accessible. And we teach them some cool web-based data science applications like CodeApp. Educators and students then use what they've learned about GIS to explore their data. We meet for one last time in April for one Saturday to focus on community engagement and science communication. Educators and students then develop deliverables to educate their peers and their communities about the risk of lacrosse, along with steps they can take to reduce the likelihood of contracting it. Then we repeat this entire process as part of a 3-year grant. I want to take a few moments and share the students' actual voices about this project.
People are larvae.
And those are mosquito eggs?
No.
Yes.
The people in the larvae are in the water.
For the Asian tiger mosquito. It's an invasive species that can cause lacrosse encephalitis.
Every Tuesday we come out, change the water, and take the paper thing, and then we send them to UT.
Things that like worms, that look like worms.
Yeah, that are tumbling around, those are the things.
I really am a strong believer in doing the science. Scientists, they don't just read about things, they're constantly going through different practices, whether it's engineering or scientific practices, hands-on, experimenting, asking questions, looking at data. So if you just read about it, it just kind of comes in one ear and out the other. When you do the science, it's something that you won't forget.
At this point, I'm going to turn it over to Corey, who's going to give you some details on the data that's collected by the students, as well as how they're using that in their scientific research.
Hey everyone, I'm Corey and I'm going to be finishing out our presentation. And I wanted to begin that process by just pointing out how simple mosquito surveillance can be. You can do scientifically accepted mosquito surveillance using just the materials that are in this image, including plastic cups, pieces of paper, maybe a marker for labeling, and some paper clips. And it's for that reason that we're able to have a community-driven mosquito surveillance project, because all these materials are safe and simple and cheap, easy to transport. So we can show educators how to collect mosquito eggs, and they can also teach their students how to do it, which is great because it lets the students get involved and have a high impact learning process, hopefully. But it also allows us to get a lot of data back out of, doing this community engagement project. And what you're seeing here is a picture of a room that we've committed entirely to raising mosquitoes from eggs to adults. So what's happening here is that the educators and students are setting out all of these traps to collect mosquito eggs. And these pieces of paper that are in all these containers, you can see an enlarged image here, they're covered in these little black dots. And those are mosquito eggs that the students collected. And what we do is take plastic containers like you're seeing in the background. We put water in the bottom, put the paper into the water so that the eggs are submerged, and then those eggs hatch and mosquito larvae come out. And they will eat food in there and eventually reach adulthood. From there, we take the adults and largely I've been doing this part, identify the adults to species and sort them out into different tubes so that I can start to do some molecular work. And I get to put on the lab coat and safety goggles and begin to extract RNA. I also use the aid of this nice robot. And so what I'm doing here is extracting RNA from all the mosquitoes so that I can perform PCR and amplify lacrosse virus. So if lacrosse virus is present in any of these mosquitoes, it's going to amplify during PCR and we'll be able to see that the lacrosse virus is there. And if there's no amplification, then no lacrosse virus is there. And so all the mosquitoes that are being collected by people participating in megabites, we're getting to screen them for the presence of lacrosse virus. So all this is to say that we're getting a lot of data. Megabites is collecting mosquito surveillance data from a larger geographic scale than what we could accomplish just within our medical entomology laboratory. And this is great because it sets up for us to get a better understanding of how mosquito species distributions are different over space and time. We can get a sense of how different locations and environments influence the abundance of different mosquito species. And hopefully we can gain a better understanding of the distribution of lacrosse virus and the risk of lacrosse encephalitis because of all this data that's being collected through this community engagement project. And the good news is that the data is good data. So here's a picture showing the average number of eggs collected by Megabites participants and by myself and other members of the medical entomology lab at UT in 2020. And what we are finding is that the data they're collecting is really similar to the data that we're collecting. And if you assume that we know what we're doing in the medical entomology lab, then that means that the Megabites participants are collecting data that we can really confidently draw scientific inferences from and use it to get a better understanding of lacrosse transmission and dynamics in East Tennessee. And moving forward, that's exactly what we want to do. One thing that I would like to incorporate in our program is this simple trap that just, again, uses plastic and water, but it collects adult mosquitoes. And the reason for that is when you get adult mosquitoes, instead of just the eggs, you can make decisions or you can learn things about how the environment is impacting the mosquitoes themselves. And we think that different environments and different compositions of species will actually influence the physical size of these female mosquitoes. And we know from laboratory studies that the size of the mosquitoes impacts the likelihood of them becoming infected and of vectoring a virus. And we also hope that by doing all these RNA extractions and looking for lacrosse virus in the mosquitoes that are being collected by megabytes participants, that we can get enough data points to actually begin doing some niche modeling of lacrosse virus itself and make some sort of model that will allow us to predict where lacrosse virus is present in Tennessee and hopefully get an idea of where the greatest risk for lacrosse encephalitis occurs. So with all that out of the way, here's a slide thanking the many people who have contributed to making Megabytes possible. We will be at this meeting live, so if you have any questions, we are now going to be present to answer them. Thank you very much for listening.